Hello everyone. In this video, we are talking about smooth muscle. There's an important concept in physiology that says that the structure of a cell, for instance, matches its function. And we'll explore this concept as we move through our exploration of smooth muscle physiology. And the way that we're going to talk about smooth muscle is to build on what we already know about skeletal muscle. So by the end of this video, hopefully we'll have a better understanding as to how smooth muscle cells are similar to skeletal muscle cells and also how they're different from skeletal muscle cells. So this is a slide that we've already seen, and this is only a reminder that smooth muscle is one of the three types of muscles that we have in our body. We've talked already at length about skeletal muscle. We will talk about cardiac muscle in the winter semester, and the next few lectures focus on smooth muscle. And one of the key characteristics of smooth muscle is that they are non-striated muscles. And we can see that if we look at the electron micrograph or the cartoon in smooth muscle. You don't see the vertical bend here as you would do here. And remember that this is because there are no sarcomeres in smooth muscle. So we still have actin, we'll still have myosin, but they will be organized differently than what they were in skeletal muscle. Smooth muscle play an important role in maintaining homeostasis, and that's true in many tissues and many systems in our body. And that's because you can find smooth muscle cells in varied locations in our body. For instance, we have vascular smooth muscle, and these are the smooth muscle that line your blood vessels. So their function there is to constrict and dilate blood vessels, and that will ultimately control blood distribution and blood pressure. There's also smooth muscle cells in our gastrointestinal tract. And when these smooth muscle contract and relax, that helps control the processes of digestion and absorptions of nutrients. We also have smooth muscle lining our urinary tract. Your bladder, for instance, will be able to relax and expand when it's full of urine. And there's also smooth muscle cells that form sphincters that will then control micturition, or basically control when we urinate and when we don't urinate. We have smooth muscle in our respiratory tract that will contract and dilate airways and therefore control airflow. We have smooth muscle in our reproductive system that can contract and relax both the uterus in females and the penis in males. That helps control reproduction. And finally, we have smooth muscle in our eye that help change the shape of the lens and therefore participate in vision accommodation. So as you can see here, we have smooth muscle in many different systems, many different tissues in our body. Now let's take a step back and think about our skeletal muscle. When we describe a skeletal muscle cell, it didn't matter whether that cell came from the biceps or your quadriceps or a muscle in your big toe. All these cells were the same. In smooth muscle, that's different. And that's because these smooth muscle cells have widely different functions. So the smooth muscle in your eye, for instance, that controls the shape of the lens, needs to have different characteristics than the smooth muscle in your blood vessel that controls blood pressure. So because smooth muscles have different functions based on where they are, their structure will also vary based on their location. So the smooth muscle in the eye has a different function than the smooth muscle in the blood vessel. That means that these two cells will also have different structures. And that's our concept of structure matches function that's so important in physiology. Now that we know that smooth muscle cells are different from one another, let's compare them to skeletal muscle. So the tagline here is that smooth muscle must be able to do different things than skeletal muscle. And here are a few examples. Smooth muscles must be able to contract over a wide range of lengths. Take your bladder, for instance. Imagine that your bladder is stretched almost to its maximum because it's full of urine. So all the smooth muscle cells that line your bladder are stretched almost to their maximal capacity. Well, it is important for this cell to still be able to contract so that you can expel this urine in the external environment. It'd be very inconvenient is when your bladder is as full as it can be, it wouldn't be able to constrict. And if you remember the tension-length relationship that we talked about with skeletal muscle, This is not true of skeletal muscle. Skeletal muscle cannot contract over a wide range of. They really need to be into this optimal starting position in order to create tension. Smooth muscle must also be able to contract in many different directions. And once again, that's different from skeletal muscle that only contract along their longitudinal axis, along their long axis. And finally, smooth muscles must be able to sustain contractions for a much longer time without fatigue. So once again, I want to highlight that when the function of two cell types are different, their structures will also be different. So let's do a quick comparison here between smooth muscle and skeletal muscle. So we have the information about skeletal muscle on the right, and we're looking to fill out the information about smooth muscle. So the first question is, what do the cells look like? Smooth muscle cells are actually much smaller than skeletal muscle cell. They only have one nucleus, and because they are smaller, they do not need T-tubules. So remember that the function of the T-tubules was to bring the action potential closer to the sarcoplasmic reticulum where calcium is. And it's really important in a big cell like a skeletal muscle cell, but it's not necessary in a smaller cell like smooth muscle. Second question, how are myosin and actin organized? So we already know that in skeletal muscle we have sarcomeres, and we already know that in smooth muscle cells we have no sarcomeres. One of the things we haven't mentioned about skeletal muscle is the ratio of actin to myosin. And you can see here that in skeletal muscle, for every molecule of myosin, it has 3 molecules of actin. The ratio in smooth muscle cell is different. For every molecule of actin, it has 12, sometimes up to 15 molecules of actin. So there's many more filaments of actin that are around the myosin. And what this does in smooth muscle cells is that it's way easier for the myosin to grab onto an actin molecule and form a cross bridge because there's just way more actin available around. Third question, how long is a muscle twitch? A muscle twitch in smooth muscle is much longer than in skeletal muscle. In some cases, one twitch can actually last up to 5 seconds. So much longer than the 20 to 200 milliseconds we have in skeletal muscle. Next question, do the cells use cross-bridge cycling to produce force? The answer here is yes. So remember that 5-step process where we describe how myosin and actin interact together? It has the ATP binding and the myosin head swiveling and so on and so forth. So this is actually something that smooth muscle and skeletal muscle cells share. So that's cross-bridge cycling is comparable between the two types of cells. Next question, how fast is the myosin ATPase in one cross-bridge cycle? In smooth muscle, the answer is slow. So one cross-bridge cycle is much slower in a smooth muscle cell than in a skeletal muscle cell. Next question, which arm of the nervous system regulates these cells? Smooth muscle cells are under the control of the autonomic nervous system. So remember, that's your sympathetic nervous system and your parasympathetic nervous system. And what this means is that we have no voluntary control over smooth muscle cells. If I ask you to dilate or constrict your airways right now, or dilate or constrict your blood vessels, you have no voluntary control over that. Final questions. How are neurotransmitters delivered to the cells? The key word here is by varicosities. We briefly mentioned varicosities when we talked about the nervous system, and we will revisit them in a few slides. So hopefully this summary table can help you conceptualize how smooth muscle cells are similar in some ways and different in many other ways to skeletal muscle cells. So now we're putting more details around these concepts. So let's look at the anatomy of a smooth muscle. So I want to bring your attention to the cartoon on the top left here. What we're looking at here is 2 smooth muscle cells, cell 1 over here and cell 2 over here. And you see a number of structures here. First, there's structures that we already know. Myosin is represented here in purple, and then actin is represented here in red. So smooth muscle cells still have myosin and actin. What is new here are those purple round shapes, and they are called dense bodies, right here. Dense bodies are the structures where the actin attaches. So if you look at the actin in red here, it anchors on the dense body on each side. Now, the dense bodies work with the intermediate filaments. The intermediate filaments are the thin purple lines that you see here. I will actually highlight them in green here, or a few of them in green. So these are called the intermediate filaments. When you take the intermediate filaments and the dense bodies together, that provides the cell's cytoskeleton. Basically, this is what will give a smooth muscle cell their shape. That's also what's going to shrink and stretch as the cell contracts and relaxes. Now, let's shift our attention to the bottom cartoon here. So what we see here is the actin filaments, as we know, in red, and the myosin filament in the middle here. There's one thing that you should recognize that's different from skeletal muscle. The myosin actually has heads throughout its molecule. When we describe the structure of the myosin in skeletal muscle, we drew it like this. So we had heads of the myosin on either side of the molecule, but there was a part in the middle that was not covered by heads. So that's in skeletal muscle. Now it's different in smooth muscle, as we just said. The entire myosin filament is covered with myosin head. What is the reason for this? What is the benefits for that? Well, remember that to create tension, we need the interaction between actin and the head of a myosin. So the more myosin heads are available, the easier it is for myosin to grab onto actin or to interact with actin. So basically, this increases the likelihood of cross-bridging happening. Now, this is especially important in smooth muscle cell because you have to realize that a smooth muscle cell can be stretched, can be contracted, and it can also take all kinds of different shapes. So what I'm trying to say here is that the actin and the myosin aren't like always nicely organized in sarcomeres like in skeletal muscle. It's a little bit of a free-for-all. So the more myosin heads that you have, the higher the likelihood of forming this cross-bridge. And this is what we see right here. So here we have a smooth muscle cell that's relaxed, and here we have a smooth muscle cell that's contracted. So the first thing I want us to notice here is that you still see the dense bodies represented by the dark dots here that are linked by the intermediate filaments, and that provides the skeleton of our cells. And when cross-bridging happens, actin and myosin pull on these dense bodies to bring them closer together. However, that can happen in many different directions. Now, these forces can come in all sorts of different directions. So we can have actin pulling in all kinds of directions that aren't necessarily always the same. As a result, the smooth muscle cell deforms. And now that's when the high actin to myosin ratio, about 12:1, and the fact that myosin has heads throughout its filament really helps because it means that myosin can interact with actin even when the cell is contracted, deformed, or even very stretched. And this is just a reminder that this is very different than what happens in skeletal muscle. This is a figure from our skeletal muscle lectures, which refers to the tension-length relationship. And when we described that when the sarcomere is very, very stretched and the actin and myosin don't overlap, it's impossible for the cell to create tension. And we get tension at 0 here. The other end of the spectrum is when the sarcomere is very, very compact and there's too much overlap. In this case, you have another problem, but you have the same result. The cell cannot produce tension. In smooth muscle cell, because of the adaptation we just talked about, we don't have that problem. Smooth muscle cells can create tension regardless of their starting length as well as their starting shape. Another key characteristic of smooth muscle is that they can sustain contractions for very long periods of time. There are smooth muscles in our bodies whose default state is to be contracted. The sphincters at the bottom of your esophagus, for instance, is always closed except when we eat. And that prevents acid from the stomach to get back up. That basically prevents gastric reflux. The sphincter in your bladder is similar. It's always contracted and only relaxed when necessary, basically to allow urine out of the bladder. So now we're talking about smooth muscle cells that sustain the contractions, not for seconds, not for minutes or hours, but for days and days at a time. So what kind of adaptations allow smooth muscle cells to sustain these lengthy contractions? Well, the first one is that a smooth muscle twitch is longer than a skeletal muscle twitch. So where skeletal muscle here is represented in red. So depending on whether we're talking about type 1 or type 2X fibers, it's going to differ a little bit. But a skeletal muscle twitch can be about 20 milliseconds. Now the smooth muscle here is represented in green, and one twitch can last up to 5 seconds. Now remember that every time there is a twitch happening, it requires energy, or ATP. So the longer the twitch means that you need less twitches in order to sustain contractions for a long time, and therefore that cell will require less energy to sustain that contraction. The second adaptation is that smooth muscle can actually slow down the activity of the myosin ATPase. So remember that the myosin ATPase is the enzyme in the cross-bridge cycle that allows the cycle to continue. Every time there's a cycle, it requires one molecule of ATP. So it's the same idea here. If you slow down the cycle, you use less ATP. So that ability that smooth muscles have to decrease the activity of myosin ATPase just slows everything down and therefore decreases ATP requirements. Smooth muscles can be classified based on their contraction pattern. We call them either phasic smooth muscles or tonic smooth muscles. So as you can see here, the phasic smooth muscles are cells that will cycle between contraction and relaxation. And there's a few different types of those cells. But the key characteristics or the key resemblance between these two examples here is that the default state of that muscle is to be relaxed. So in our first example right here, the smooth muscle is normally relaxed and only contracts when there's a stimulus. So it's relaxed, contracts when there's a stimulus, and goes back to its relaxed state. We have smooth muscle at the top of our esophagus that follows this pattern. Now, the second example is smooth muscle that will alternate between a relaxed state and a contract state. Relax, contract, relax, contract, relax. An example of that is smooth muscle in your intestine. When there's food in your intestine, your smooth muscle will go through these cycles of relaxation and contraction, and that will actually help mix the food that you just ate and then promote digestion. So these phasic smooth muscles really cycle between the contraction and relaxation, but their default state is relaxed. Now, tonic smooth muscles are different. Their default state is to be contracted. One way that we can remember that is that the word tonic refers to tone. So these are smooth muscles that always have some tone or some contraction to them. Once again, we have a few examples here. In the first example, we've already mentioned that it could be the sphincter in your urinary bladder. It's always contracted, preventing you to pee in your pants, for instance. But when you do actually want to urinate, it relaxes, lets the urine out, and then goes back to a contracted state. In the second example, we're looking at a smooth muscle that's contracted, but the amount of tension in it varies. So more tension, less tension, more tension, less tension. So it's still continuously contracted, but the force of the contraction can vary. Smooth muscle cells in your blood vessels are a great example of tonic smooth muscles. The other way that we can classify smooth muscle is based on their communication pattern. Once again, we have two categories: single unit smooth muscle cells and multi-unit smooth muscle cells. Single unit are by far the most common one. These are smooth muscle cells that are electrically linked together. And the main advantage of this structure is that it allows electrical signals to travel very quickly from the cytoplasm of one cell to the cytoplasm of the next cell. Let's look at the cartoon on the right here, which represents a single unit smooth muscle. So we have a number of cells here: 1 cell, 2 cell, 3 cells, 4 cells, and so on. The first thing I want to highlight is that the sarcoplasm of each cell is linked together via gap junctions. So we have gap junctions that are represented here in the cartoon. Basically, anywhere you can see arrows, I'm just highlighting them in green here. So these are gap junctions. And we've talked about gap junctions already in this course. They are channels that allow small molecules like sodium, calcium, potassium to travel through them. The implication of this is because the signals can travel very quickly from one cell to another, to the neighboring cell, and so on, is that all the cells will contract at the same time, and all these cells will also relax at the same time. And that's why they're called single unit smooth muscles, because they act as one unit. And this is a great characteristic when you need a number of smooth muscle cells to coordinate an action. The other point where I want to draw your attention here is how these cells receive the signals. So in blue here, we have the varicosities that will deliver the neurotransmitter. These are varicosities. Deliver the neurotransmitter. Now, one of the things that you can notice is that these varicosities are only located close to the superficial layer of the cells. The neurotransmitter here are those green dots, and you can see that the neurotransmitter binds to receptor on this cell because it's on the first layer. It binds to receptor on this cell because it's also on the superficial layer and this cell. But the cells that are deeper, this one, do not directly receive input from the neurotransmitter. Now, does it matter? Absolutely not, because we know that as soon as a superficial cell is activated, the ions will be able to travel very, very quickly through all of the other cells, and they will all have the same behavior. So as I said at the beginning, this is the large majority of our smooth muscle cell that work this way. The other type of smooth muscle cell is called a multi-unit muscle cell. And this is when cells are not linked together, and therefore each cell must be stimulated independently in order to contract or in order to relax. The best example of that are the smooth muscle cells located in our eye. And that's because when our body can control every cell individually, it allows for a very, very fine control of contraction within a tissue. And that's what you want for smooth muscle that dictate the shape of the lens, where, minute adjustments will make a big difference in our vision. So what we see here with those multi-unit smooth muscle cell is that, first of all, there's no gap junctions, right? Every cell is fully independent from the other. And the other thing that we see is that these varicosities actually go all the way through the tissue in order to stimulate every single cell independently. Now, going back to these single unit smooth muscle cells, the one that we have a lot of in our body, We talked about how they're electrically linked together, but we haven't talked how they're mechanically linked together. The electrical link is via the gap junctions that are highlighted here in blue. That's where the ions can pass from one cell to the other. But what I want to highlight is the mechanical coupling right next to it. So there's connective tissue between two smooth muscle cells that will allow the transmission of force between the cells. In other words, if one cell contracts, it can pull its neighbor in the same direction. And that's another characteristic that helps smooth muscle cell act as a single unit. The last thing I want to highlight about smooth muscle is how they receive information from the autonomic neuron. Once again, let's take a step back and think of our skeletal muscle. In skeletal muscle, you needed every single skeletal muscle cell to receive information from a motor neuron. So that requires a lot of neurons. In smooth muscle, many smooth muscle cells can receive information from the same autonomic neuron. Why? Because of the varicosities. So varicosities, and you can see them right here, are basically part of the axon that is swollen. So if I bring your attention here, we have the postganglionic neuron here in its axon. And when it gets closer to the target tissues, it starts having all of those little beads almost, but there's swelling of the axons that we call varicosities. And inside the varicosity, you have the neurotransmitter. And you can see that the axon branches in all sort of different directions, and there's varicosities throughout the target tissue. And this structure allows one postganglionic neuron to affect a large area of the target, in this case, our smooth muscle cell. So this is it for this video. Hopefully we now have a better understanding of how skeletal muscles are designed and what are the benefits of being designed that way and how they differ from skeletal muscle cells.